Hi everyone, welcome to the Module 5 labs. We're gonna get the wonderful beads out again and do some work with population genetics. So go ahead and get to experiment 1, which is genetic variation. And I have my combined lab report printed out here to kind of walk you through what to do. So the first step it tells you in the procedure is to pour and mix 50 blue beads and 50 red beads into a beaker. Okay, so these are all mixed up. And then take 50 green beads and 50 yellow beads and mix them up into another beaker. So we have these two ready to go. And then what they want you to do is, without looking, kind of similar to what you did in Module 3, you're just going to pick 50 beads out. So here, and put them in another beaker. And we're going to have this be beaker 1. So I'm just going to start selecting. So 2, 4, 6, 8, 10, 12, and you don't have to watch this whole thing, so I'll come back after I've selected. So I'm going to select 50 out of this one and 50 just random beads out of this one. Okay, so I'll come back when I've done that. Okay, so now that I have random beads, so I just pulled 50 beads, 50 random beads out of here without looking, and I put them into beaker 1. And then I pulled 50 just random beads out of this one and put it in beaker 2. So now you're on your part 1 post-lab questions, and it asks you, what is the initial gene pool colors of beaker 1? So how many red beads and how many blue? So you have to take out, pour them out, count up how many reds you have, count up how many blues, and write them down. And then same thing with this one. Pour them out, count how many yellows you have and how many greens. And then you're going to do a calculation to figure out the gene frequency. So typically the calculation is out of however many you have. So if you had 22 red beads, you would put 22 divided by 50, and that will give you the gene frequency of red. If you had 25 blue beads, you'd put 25 over 50, and that would give you the gene frequency of blue. And then you're going to just find the gene frequencies of each color. When you have counted them all, put them back into the beaker and keep them, because you're going to need them for part 2, which we're going to go to next. So you answer all your questions, and then you go on to part 2. And in your instructions, it tells you, it says dump the contents of beaker 2 into beaker 1 and mix thoroughly. So here we go. Okay, and I like to kind of do like a maraca mix, so that way they don't go anywhere. All right, so that's pretty mixed, and one's still in my hand. And then it says pour approximately half the beads from beaker 1 into beaker 2. So this does not have to be exact. You're not having to count out little beads, just pour. And then look, that's not half. Okay, that's about half. And then now you're going to use beaker 2 to answer your questions for your post-lab in part 2. So now it's asking you, what's the new gene pool? So, take out your reds, count them. Take out your blues, count them. Take out your yellows, take out your greens, count them. That'll give you your gene pool. Then you have to find the frequency of each color. So, you have beaker 1 that you're going to have to count. Then you're going to have beaker 2 that you have to count. And then you'll go through and calculate the frequencies. And then you're going to just be looking at the variation. So that's kind of part 1 for genetic variation. Keep these beads as they are in the beakers for the next part. All right, so now we're on experiment 2 called genetic drift. We kept the mixed beakers and beads from experiment 1 to do genetic drift. So what you're going to do here is it tells you to randomly remove half of the beads from beaker 1 and half of the beads from beaker 2. eyeball it. Okay, it doesn't have to be precise. And what this is representing is like a destructive event. So like a wildfire or a hurricane that comes in and just wipes out half the genes. So we're going to go ahead and randomly remove half. I'm just going to set it aside. Randomly remove half. Set it aside. And so now we're going to answer our post-lab questions. And what will happen is you're going to repeat this 4 more times. So a lot of people like to make a table of this when they do this. That's totally fine. So like for instance, this would be considered round 1. And so in round 1, you would say, okay, beaker 1 has however many reds, however many blues, however many yellows, however many greens. And then beaker 2 has X number of yellow, red, blue, green. And then what you do is put all the ones that you took out back into the beakers. And then do it again, right? So remove half. Remove half. Fill in your data table. Okay, put them back in. Do it again. And then you just repeat it 4 times and do your post-lab questions, and that's it for genetic drift. Then you're keeping the same beakers for your experiment 3. So put everything back and then get ready for experiment 3. Hi everyone, so we're on experiment 3, stochastic events. We have our beakers from the previous 2 experiments. And so following the directions, we're going to consider this population 1 and population 2. So we're looking at genes in a population. And just like in real populations of animals or humans, you have a variety of genes in that population. Some genes can be more dominant than others or more prevalent. Some genes can be less prevalent, just depending on the population. So if you look at your instructions, it says randomly select 10 beads from beaker 1 and place them in an unused beaker. So I'm just going to randomly select 10. Okay, so what I ended up with is 2 greens, 3 yellows, 4 reds, and 1 blue. So you can see that the dominant gene is red. The one that's more prevalent, not dominant as in like whenever we did genetics, dominant, recessive, but which one is more prevalent. And then the blue is less represented. There's only 1 individual with blue. And so you're just going to compare what you have here with what you have here. So this is basically showing you that when a stochastic event happens, right, less gene pools in that population are represented, because if you compare it to the original founding population, you can see there's a lot more of each gene color, right? Like there's more greens, there's more reds. And so you're just going to use that to help you answer your post-lab questions. We're going to put these back in here. And then this time you're going to select 10 from this beaker. And same thing, you're just going to compare with the original population. Try not to look when you're doing it because you want it to just be random chance. Okay, and so you might have differences in, what you had from beaker 2 than what you had from beaker 1. And so you're just kind of noting that and how chance can affect a population, or destructive events can affect a population. All right, everyone, we are on experiment 4, which is natural selection. You're going to need all of your red and all of your blue beads, and a beaker, and your combined lab report. So you're also going to need to print out these 2 pages, the red habitat and the blue habitat. Shouldn't cost you that much money. I've had people try to do it on their laptops or their tablets before, and it just doesn't work as well. So you really need to print them. I printed all of my all of the lab instructions and all of the combined lab reports at Staples for like $20. So it's not too bad, but I do understand that some people have, economic issues that cause you to not be able to do that, and that's fine. You can always go to a library, and a lot of times they will print pages for you for free. So you can do that as well. So what the directions ask you to do is put 50 red and 50 blue beads into a beaker. And you're going to mix them really well, and you're going to pour them over the red habitat sheet. So we're just going to pour, and see, they go everywhere. So if they go off the paper, just kind of shove them back onto the paper. Okay. And then it says for you to return to the beaker the beads that fall onto the habitat area that match their color. So if a red bead falls onto a red area, return it. Now here's the tricky part. For each one you return, you have to add another one. So I just pulled these 2 red off of a red area. So I have to go into the red bag and pull out 2 more red beads. Okay. So I have some blues that landed on a blue area. So I have 2 blues that landed on a blue area. I'm going to return those and add 2 more blues. And so that's all I'm going to do, is I'm just going to go through and pick out all the beads that landed on an area that matches their color and give them more beads. Now the reason why you're doing this is because it's assumed that red is going to blend into red, right? They're going to be camouflaged. And so they're going to survive and have offspring. So the ones that I'm adding is the offspring. So I'm putting these back in there. Okay, and it'll take you a while, and it's kind of tedious, and I apologize for that. Just do the best you can. Okay. And so eventually you're going to get to where you have your whole population back in there. And so the directions then tell you that you're going to repeat this 3 times, and you're going to record the distribution of colors. So essentially, after you've gone through it 3 times, you're going to have a certain amount of red and a certain amount of blue left in your beaker. You're going to take them out, and you're going to count. So in your results, where it says experimental results, you're going to add up how many blues ended up being saved and how many reds ended up being saved after you've done it 3 times. Then you're going to put them all back in the beaker again and go to your blue habitat page, which is right behind this one, and do the same thing on the blue habitat. If blues land on blues, take them out, add blue beads to it, put them in the beaker. If reds land on reds, take them out, right? They mate, they have offspring, give them more beads, and then you'll record on your experimental results how many blues and how many reds on the blue habitat. And so that's just kind of showing you natural selection with beads. Hi everyone, we are on experiment 5 for module 5, the sickle cell anemia inheritance patterns. So in this particular one, you're going to be looking at sickle cell traits, and we're going to be using red and blue beads. So in one beaker, you're going to put 25 red, which represents the normal dominant allele, and then 25 blues, which represents the sickle cell allele. And you're just going to mix them really well. And then it says, without looking, remove 2 beads and repeat that 10 times. So I'm just going to put them, whoops, yeah, it's messy. So I'm just going to put them in a row here. Until I get to 10 times. So you can see it's just random, so yours are going to be different than mine. Okay, so I've removed 10 pairs. Now it says to record whether the beads are homozygous dominant, heterozygous, or homozygous recessive. So there's a table that you will put that you can record your data with, and then it says assume that each individual with 2 lowercase s's genotype passes away. So in my 10 that I pulled out, I had 2 that were homozygous recessive. Okay, so we removed them from the population, and the remaining beads survive and reproduce. So what that means is that you will, kind of similar to your natural selection one, for every red bead you have to add another red bead, and for every blue bead you add another. So I had 3 blue beads, so I just add, and you can either get this from your bag or just have a beaker of red and blue beads. So I need to add 3 more blue beads to my population. And then I had 2, 4, 6, 8, 10, 12, I had 13 red beads survive, so I'm going to need to add 13 more red beads to this, so I'll have 26 red. And then I'll put them all back into my my beaker, my original beaker, and I'll do it again. So I'll need to add the 23 reds. Or sorry, the 13 reds, not 23. Okay. And then I'll just, mix it up again, and then pull another 10. And just doing the same thing, right, where if you have 2 blues that come out, then they disappear from the population. And you just repeat this pattern 7 times, and you just, as you're doing this, pay attention to your post-lab questions, because it starts asking you, like, what are your observations? Like, what are you seeing happening to the red beads? What's happening to the blue beads? What's happening to the sickle cell trait as you go through this? And that's it. That's pretty simple to work through.